When we talk about proteins, we're actually talking about something known as a polypeptide. Poly is many. Peptide is the monomer for proteins. So when we look at these guys, we're actually going to take an amino group that we saw before in our functional group. We're going to add a carboxyl, and we're going to add a functional group. This is the basic monomer for every single protein in your body. So when we look at these polypeptides, I mentioned to you before that we're going to use 20 amino acids to build them. And they're going to be involved in about every function in your body. So function is going to be highly variable based on their shape or form. And we look at each of your cells' mass as being about 50% protein in your cells, in cell mass. So this is a lot. We've got to think about a lot of things with our proteins. This is a basic amino acid structure. The amino group, as I mentioned, the carboxyl group all around this central carbon, and then this R. R is equivalent to functional group. No, I don't know why it is R, sorry. But which component of this would determine whether or not an amino acid is hydrophilic or hydrophobic? Well, you could say, all right, these oxygens are out here. Maybe they would be hydrophilic. Water tends to like those big oxygens. But what we're actually going to look at is this R is the most important component for determining whether or not this amino acid is going to be hydrophilic or hydrophobic. This is why. If the amino acid structure is all the same and we're going to stick these amino acids together to form large proteins, large polymer groups, fine. What we're really interested in is this thing that sticks off the bottom. This is the R group we mentioned. And in this case, all carbons, hydrophobic. It's not going to interact with that water at all. But then we've got oxygens here, hydrophilic. So this is good with the water. Works very well. This becomes extremely important when we start putting our amino acids together through a dehydration reaction, just like we build everything else. If you're building, you're doing a dehydration reaction. There is no choice. And when we do this, we're actually going to create something known as a peptide bond with those amino groups. The peptide bond is how we get the term polypeptide. So it's many peptide bonds that hold this protein together. So we take two amino acids, we join them together through a dehydration reaction. In this case, now we have a dipeptide or two peptides put together. Fine. Okay. But when we look at a polypeptide, we could be looking at hundreds or thousands of amino acids all put together. The critical part here is order. How are they put together? Which amino acids are in which sequence? So we said we have 20 amino acids to work with. But of those 20, maybe you put 1 and 2 and 4 and 1 and 3 and 4 all in a line. And if I change this, this is different than this. Think about amino acids like letters of the alphabet. You can put them together in all kinds of sequences to build different words. In this case, we're building different proteins that are going to do different jobs. They can catalyze reactions. What does it mean to catalyze a reaction? Makes it go better. It can regulate things in your body. It can build muscle, right? There are all kinds of things that proteins are going to be able to do for us. So this is just a small list of all the things that proteins are doing in your body, from storage and transport to structural, to receptors and signaling, to defensive. We'll look at some of them associated with your immune system later on. There are just tons and tons and tons of jobs for this combination of amino acids put together into this polypeptide chain. Now, as a chain like this, it has no function. A long line of amino acids isn't going to do anything for us. What we have to think about are those functional groups again. Why did I tell you functional groups were so important? We said the functional groups are important because of the hydrophilic and hydrophobic parts of the R group or the functional group. Why are they so important? Because this protein line chain is going to fold. And when it folds, it's going to fold in your body when it's exposed to water so that all of the hydrophobics are on the inside and all the hydrophilics are on the outside. And when it folds, it's going to form this groove or something known as an active site. This will be a critical piece of our enzyme discussion and how they actually function. The problem is proteins are easily altered by the environment in your body. Okay, so temperature, pH, salt, all of these things can change how that protein is folded. We go through what we call protein denaturation. It essentially means it falls apart. So if the protein falls apart, it can't do its job. That list of things that we talked about, it's history. It's not going to do any of them if it falls apart. The primary structure is the amino acid chain. It's the first thing you make. And then we fold and fold. The tertiary structure could give you a functional protein. And a quaternary structure is 2 tertiary structures or more put together. So a quaternary protein would be huge. And you have some of them in your body. Some of them are very, very large. Okay, so here's the basic gist. We start to fold this. It's going to fold based on water. And it's going to be based on the love or hate for water. So hydrophilic or hydrophobic. All of the hydrophilics in your body will stay to the outside. The hydrophobics fold to the middle. I mentioned to you these quaternary structures can be very, very large. One of them in you that we see a lot is collagen. Where do you have collagen? Well, I tell you here it's in tissues and bones and ligaments. But if you pinch your skin and pull it up away from your muscle, do it gently, it kind of hurts. The amount of elasticity that your skin has that allows it to go back into place is your level of collagen. If you have your grandma do the same thing, her skin doesn't snap back in place quite as easily. So that's collagen. Over your lifetime, it breaks down. It doesn't work as effectively. It goes through what we call denaturing. So we take our primary structure, our quaternary structure, and so on, and if we're going to denature this, essentially we're going to reverse the process. This denatured protein is the broken down protein because of environmental conditions, and we essentially cause this protein to fall apart. Our nice quaternary structure is lost, and we eventually get back to this amino acid chain, which is non-functional as a protein. Now, we talked about large carbohydrates, we talked about large proteins. Fats are a moderate size, like we talked about, but many of the things that we're going to see are actually what we call combo or combination molecules. So lipoproteins, I mentioned this to you, HDL and LDL. High density lipoprotein, low density lipoprotein. What is a lipoprotein? Lipo is fat, protein is protein. So it's a fatty protein. What about a glycoprotein? Glyco is a carbohydrate plus a protein. We see them on the surfaces of your cells. These green chains are carbohydrates attached to this brown blob. The brown blob is a glycoprotein. It's extremely important on the surface of your cells, and we'll look into those when we get to cell structure.